All right, so for our first example, we have a complete autosomal problem. It says in garden peas, smooth seeds are dominant. We're going to use a capital R for that. Wrinkled seeds are recessive, and we have a purebred pea plant that has smooth seeds. So first of all, we do want to identify those parent genotypes. So it says if we have a purebred pea plant that produces smooth seeds, then we're going to have a homozygous dominant parent. So big R, big R. Then we have a pea plant that is homozygous recessive. So that means that that parent would be little r, little r. Then we can draw the Punnett square. And when we draw the Punnett square, we can see that all of the genotypes of the offspring would be the same. There would be a 100% heterozygous genotype for their offspring, which also means that there would be 100% of their offspring would have smooth seeds. For the next problem, we have another complete autosomal trait. So again, that's on the 22 non-sex chromosomes. And we have freckles as being the dominant trait. We have a man and a woman who are both hybrid. So hybrid means that they are heterozygous for those freckles. Both of them have one dominant allele and one recessive allele. So if we draw a Punnett square for that particular trait, the probability of them having a child that is homozygous dominant, we can see that one of the four boxes would have two dominant alleles, homozygous dominant. So 25% chance of being homozygous dominant. And then we can also see that there is a 25% of being homozygous recessive for the trait. Okay, for number 3, guinea pigs, black fur is dominant and white fur is the recessive trait. And it says that we've got a white female that is mating with a hybrid. So that means that the male is heterozygous. So we have a male who is capital B, lowercase b, and we have a female who is lowercase b, lowercase b. And when we put that together with the Punnett square, we can calculate the probability of having black fur would be about 50%, because we can see 2 of the boxes will be heterozygous and 2 of the boxes will be homozygous recessive. And so we're going to have a 50% chance of each of those genotypes, and only the heterozygous one is going to have the black fur, while the homozygous recessive one will have white hair. So the next problem says we have guinea pigs and the dominant allele is capital A, and that's going to produce short fur. The recessive allele, lowercase a, produces long fur. So if we think about this, that means there's 2 different possible genotypes to have long or short hair. Capital A, lowercase a, and 2 capital A's. This one says that the male is heterozygous. So the male is going to be capital A, lowercase a, and has short hair. That male fertilizes or mates with a homozygous recessive female. So that is going to be lowercase a, lowercase a. And when we create that Punnett square, we can see that we will have 2 boxes that are heterozygous, capital A with a lowercase a, and we will see that there are 2 boxes that are homozygous recessive. So that means we have about 50% chance of having the short fur, that would be the heterozygous offspring, and we have a 50% chance of having long fur, that would be the homozygous recessive offspring. So when you're doing Punnett squares, if you have 2 individuals that are both heterozygous, which is what a hybrid is, it means that it is heterozygous for the trait. That means 1 dominant allele and 1 recessive allele. So anytime you have a Punnett square that has 2 heterozygous individuals mating, you're always going to get the same results. You're going to have 1 box that is homozygous dominant, 2 boxes that are heterozygous, and 1 box that is homozygous recessive. So the possibility of having an offspring that is homozygous dominant is going to be 25%. So here we see the question the opposite way. It is asking us what would be the genotypes of the parents. But again, the clue is seeing that 25%, 50%, 25%. If we were to put these results into a Punnett square, we can then deduce what the parents would be like. And in order to get this result, you would have to have two heterozygous parents. Next, we get into some examples that are incomplete dominance. The way to know that this is incomplete dominance is because when you have a red flower and a white flower, we don't get the dominant trait of red. We get a totally new trait, and that trait is pink. And so the heterozygous individual, the fact that there are three different traits, helps us know that this is incomplete dominance. So if we have a white plant, that is going to be little r, two recessive alleles, and we cross that with a pink plant, which is going to be heterozygous, big R, little r, we are going to get 25% are going to be pink. 50% are going to be pink and 50% are going to be white. So the probability for producing white phenotype for these plants when we have this cross is a 50% chance. This next example is also incomplete dominance. Again, how can we see that? Is that we have three different phenotypes. We have red, yellow, and chestnut. So if we have a chestnut horse, that means that that individual is going to be homozygous recessive, little g, and we're going to mate that with a red horse, which is going to be homozygous dominant, big G. When we put that together in a Punnett square, we can see that 100% of the offspring will be heterozygous, and the heterozygous trait is yellow. So the probability of a colt being born yellow would be 100% for this cross. All right, so here we have roan cattle. Roan is a trait that is heterozygous and it shows incomplete dominance. So when the cattle has capital R, lowercase r, so one dominant allele and one lowercase allele, the cattle is going to show the third phenotype of roan. So we need to draw a Punnett square and determine what the probability of having a calf be born red would be. So if we cross two heterozygous cattle, we see that one of the boxes will be homozygous dominant, and that's the only one that would be red. So we have a 25% chance of having a red offspring being born. The next example for incomplete dominance, we have brown rabbit fur is going to have the heterozygous trait. So that's our third phenotype. So that's why this is incomplete dominance. Red is dominant to white, and we're going to cross a white and a brown rabbit. So the white rabbit would be lowercase r, since red is dominant, and our brown rabbit would be heterozygous, so capital R, lowercase r. And when we draw the Punnett square, we're going to see that 50% of the boxes will be heterozygous, so we will have a 50% chance of having brown offspring. The other 50% will be white. On this next example of incomplete dominance, we have the white color being the dominant fur color for the cats, black being the recessive, and gray being the third phenotype we see that is heterozygous. The question asks us that two gray cats mate. So that's going to be a heterozygous crossing with a heterozygous. And anytime we have a heterozygous crossed with a heterozygous, we always get the same results. We're going to have 25% are going to be homozygous dominant, 50% will be heterozygous, and 25% will be homozygous recessive. And it wants to know the probability of having a black cat, which would be the homozygous recessive trait, 25%. On this next example of incomplete dominance, we have rabbits with some different types of fur. We have Angora fur, Rex fur, and medium length fur. And it asks us to cross an Angora with a medium fur. So we're going to be crossing a homozygous dominant with a heterozygous. And when we do that Punnett square, we are going to see a 50% probability of having the heterozygous Rex fur. And we will see a 50% chance of having the Angora fur. So it would be 50% of having the Angora. All right, so now we're moving on to another type of inheritance, codominance and multiple alleles. So codominance means that A and B are both dominant together. So if we have both the A allele and the B allele, we're going to see both of those traits, and that's how we get blood type AB. When we have the O allele, O is recessive to both A and B, and the only way that you can get blood type O is to have two recessive alleles. So in this particular example, we have type O, which means that the genotype must have two O alleles, and they are mating with a heterozygous for type B. So that means that they have one B allele and one O allele. The B is dominant over the O. When we draw a Punnett square for that, we're going to see that we have a 50% probability of having a child with blood type O, and the other 50%, the child would be heterozygous for blood type B. All right, in this codominance example, we have a woman who has blood type B and is heterozygous, and the child has blood type A and is heterozygous for that blood type. So we actually have to try to figure out what the blood type of the father would be. So we need to draw some Punnett squares and try to figure out what are the different possibilities. Well, mom being blood type B and being heterozygous, she has a B to give or an O to give. And so that means that if her child has blood type A, she could not have given the gene of the B. She had to give the O. So that means that the A had to come from the father. Well, the father could be blood type A and give an A, or the blood type of AB and give an A. So no matter what, the father has to have an A within the blood type. So either is actually blood type A or could be blood type AB to give the A. All right, so if we have a child that is born with blood type AB, then remember you have to get one allele from mom and one allele from dad. So that means that one parent has to have an A allele and one parent has to have a B allele. So you could have one parent with blood type A and one parent with blood type B. You could have one parent with blood type A and a parent with blood type AB, or you could have a parent with blood type B and a parent with blood type AB. So there's three different kind of possibilities that we can go with here in order to get the blood type child with type AB. So make sure that you've got all the different possibilities. Okay, for this example, we have blood type A with a heterozygous type B person. So we know that the B is heterozygous, but we actually don't know if A is homozygous or heterozygous. So I do have two Punnett squares here doing it both ways to determine what the probability of having a child with blood type AB is, because we don't actually know if the individual is homozygous or heterozygous for the trait. So when we do this cross, we can see that if the A blood type is homozygous for blood type A, that then there's a 50% chance in that Punnett square of having blood type AB, whereas if the individual with blood type A is heterozygous, then we only have a 25% chance of having blood type AB then. All right, so for the next example, we have Hirsch and a mother with blood type O, and we need to find out if Hirsch is possibly the father of this child. Well, baby is blood type A, and baby has to get A from someplace. So that means that the A, if mom is blood type O, she only has the O allele to give. So that means that the A that baby gets has to come from the dad. Well, if dad is blood type B, he can't possibly have any A alleles. So if he doesn't have any A alleles, then that means that can't be the dad. For this last one, we have 50% of the offspring is type AB. So that means that they must have the genotype of AB. The rest are type A. So if they're type A, they could be AA or they could be AO. So if we were to do some Punnett squares where we only end up with these particular genotypes, if we had family members who are blood type B and we have family members who are blood type A, then we could get only AB blood type and then AO. But it does not say that anyone in this family has blood type B. So we're not going to go with that as a possibility. So we're going to make sure that our Punnett squares always have blood type AB or a blood type A, or the A with the O. So if we had homozygous dominant, then we would have all blood type A's and blood type AB. If we had an AB with an AO, then we would still have all blood type A's. We'd have an AB, but then we'd get a BO. So we are going to guess that it is not A with an O. We're going to guess that the genotypes are going to be homozygous dominant and heterozygous, because we don't want to have anything with B's in it as being the blood type.